So I'd like to start off by starting with my talk on imaging patient-derived xenographs, and I'm going to talk about why this time it's personal. So what if I told you that undergoing cancer treatment should be similar to having an experienced tailor creating outfits designed to your unique measurements? Many of us have heard the term today or the idea of personalized medicine, which in the context of cancer refers to the goal of moving away from a one-size-fits-all approach to effective treatment as well as the care for patients. Unfortunately, many studies of cancer have long relied on the use of cells grown in a dish under artificial conditions to draw conclusions about how cancer progresses in the body, as well as the effects of new treatments developed. While these studies have been quite useful, these cells are actually not the ideal model for cancer research, as they are similar to wearing an off-the-rack suit. First of all, unrepresentative of our unique biological differences, and would be a poorer fit than if they were tailored to a cancer patient's specific needs. This results in poor predictive power. So what if we had better models of cancer in the lab that could not only account for the differences between individual cancer types, but also better represent our diversity? I'd like to introduce you to patient-derived xenograft models, or PDXs. PDXs are made from tissues or cells that come directly from a patient's tumor and are then implanted into an immunodeficient mouse where they could be grown, monitored, and tested for therapeutic responses. Most importantly, these PDX models retain the unique characteristics from the original tumor from the patient. They can be monitored for interactions with the surrounding cells in the body, and ultimately they would allow for more accurate and personalized treatment evaluations. My research focus then is studying a PDX model which came from a tumor from a consenting patient with triple negative breast cancer. And I use imaging technology to track how these implanted cells progress over time. The reason that imaging is our tool of choice is that it allows us to study the cancer progression as it's happening, rather than evaluating the disease progression after experiments have taken place. The first imaging modality that I'd like to introduce is called bioluminescence imaging, or BLI. This imaging modality detects light that is emitted from cells which have been engineered with the light generating enzyme. This means that only cells that are alive can generate light, making it an excellent option for tracking cancer progression and treatment response. The second imaging modality that I'll be presenting on today is called magnetic particle imaging, or MPI, which is an emerging imaging modality that detects iron nanoparticles. In our lab, we've developed ways to label cancer cells with iron so that they can be detected throughout the body. We are also the first and only lab in Canada to have this imaging modality on site. In these images, iron appears as bright hot spots, which you can see in an image of signal from a tumor, which is shown on the slide there. So as I mentioned, I'm studying a PDX model of breast cancer. This particular model has been engineered with a light generating enzyme, as I mentioned, and has been labeled with iron, allowing it to be tracked with both imaging modalities. This allows us to harness each of their complementary benefits to track the progression of this PDX model. In this image here, we can actually see the cells from the PDX shown in red, and the iron I've labeled the cells with shown in blue. This shows that the cancer cells have been labeled well with iron, as we can see around 80% of the cells contain iron inside of them. Now we can move on to the imaging. So in this image here, I've implanted the PDX cells into the mammary fat pad of a mouse and imaged it over time with bioluminescence imaging. The signal we see here is the light that's emitted from the cells, and we can use this technology to see that the cells are alive, as well as the fact that they're growing over time. Here we can see the same number of PDX cells were implanted into a mouse and imaged over time, but this time using MPI. And this is actually the first time that this modality has been used to image a breast cancer PDX. As we can see, this time the signal is actually decreasing, and this might be due to the dilution of iron with each cell division and tumor growth. So the key takeaways is that PDX models allows us to study cancer using a model that provides more clinically relevant results than using a traditional cell line. And the ability to monitor this type of model with complementary imaging modalities provides a way to study personalized cancer treatments and evaluate their success in patients. So the next question is, how do we harness the predictive power of PDX models and this work to actually create change for cancer patients in the future? This strategy is very promising for future drug development, which currently has enormous costs due to the number of failed drugs from poor predictive values of current models. PDXs are more representative of what's seen clinically, and so their use would allow researchers to predict and understand a new drug's effect more precisely. Precision and personalized medicine would also allow therapeutic regimes to be developed for specific subtypes of breast cancer, and many patients have already benefited from this therapeutic strategy. Lastly, PDX can be developed from tumors of patients in clinical trials, and this is where a drug is tested in a patient and a mouse at the same time. And this would be beneficial to study how similar outcomes are and to learn more about why a patient may resist or have a successful treatment. So in summary, I hope I've convinced you that cancer treatment is very personal. Studying patient-derived xenograph models with imaging is so key to understanding the progression of cancer and can be used as a platform to evaluate therapeutics to advance personalized medicine for cancer patients. So thank you so much for listening and for the RIOT team for this opportunity. I just want to recognize the amazing Foster Lab and the other trainees listed here, as well as my sources of funding. Thanks so much.